over to you, Nick.
Great, thank you. Thanks for the introduction. Can you hear me okay?
Yeah.
Brilliant. Okay, always a good thing to check with Zoom and so forth, particularly given I've only just joined, I'm afraid. Thanks for the introduction. I'm going to share my screen shortly if it lets me. So can you see that okay?
Yes, I can see it.
Brilliant.
Nice and steady.
Excellent. So thanks for the introduction. I'm a gastroenterologist here in Exeter. I've been here for almost 6 years, and I've been very involved in IBD research, both before I came here and in the time I've been in Exeter working with Tariq Ahmad and Claire Boucher and James Goodhand and others. I'm going to talk about Clarity. This is a study that I guess 2 years ago we had never envisaged we would be doing, but it became clear sort of early on in the pandemic that some of our normal research wasn't really going to sort of happen in the way it would while the pandemic was going on, and there were some really important questions to answer about the interaction between COVID-19 and inflammatory bowel disease, and in particular some of the treatments we use. So I'm going to give a talk through what we've been doing, present some of the data we've got as well, and then hopefully there'll be some time for questions as well. It's a relatively small group. If there are questions that come up as we go, I'm happy to try and field those as well, if that's helpful. So I've got some disclosures, and to say our funding has mostly come from industry for this project. We did try to get money from the NIHR, but we weren't successful, although we did get badged as an urgent public health study. So we've had unrestricted grants from a number of different industry partners, but they weren't involved in the study design and haven't really had any control over the results that we've generated or presented as well. So of course, COVID has been sort of dominated all of our thinking for really for the last more than 18 months. This is data that was up to date as of a couple of months ago, looking at the cases per million people. And obviously the UK has had certainly plenty of cases with its various waves of COVID, depending on how you count them. We've also, unfortunately, had quite a number of people die. This is not this isn't relative to the population, so obviously that needs to be taken into consideration. But what is evident is that the UK, although we have had quite a lot of deaths, that is much flatter now than it was earlier on in the pandemic, and that's not always the case elsewhere. And really, this has been, I think, in a large part, a result of the success of vaccination. Again, these data aren't bang up to date. These are up to date as of July, but we can see that in the early part of the pandemic in the UK, we got a mirroring of the case rate here in blue with the death rate here in red. Obviously, the scale of the numbers is different. And later on, after the advent of vaccination, we see that is no longer the case. So although there are still people dying, and I showed that on the previous graph, they're much smaller in number compared to what the case rate has been doing. And in countries where vaccination uptake has been good, you can see in Portugal as well, that's similar. In countries where there's been poor uptake of vaccination for all sorts of reasons, economic and others, we continue to see ongoing high death rates with respect to the case rates. And so vaccination has really been a success story in the pandemic, and has allowed us to obviously all the deaths are bad, but at least we've had fewer deaths than we might have otherwise had without vaccination. Inflammatory bowel disease is my normal clinical area of interest. For those less familiar, this is a chronic relapsing remitting condition. The two main forms are Crohn's disease and ulcerative colitis. Crohn's disease can affect any part of the bowel. Ulcerative colitis generally only affects the large bowel, the colon. The global prevalence is reported to be 0.3 to 0.5%. Based on our local data, we think the point prevalence at the moment around East Devon is around 1 in 140, so a bit higher than that. And it's a disease that's most commonly diagnosed between teenage years and 30s, but can be diagnosed at any point, including in people's later life. And most people will continue to live with their disease through the rest of their life. So we have plenty of patients of all age groups. It is diagnosed in childhood as well, even in very young children, although in extremely young children, it's often a sort of Crohn's or IBD-like condition that's more related to a genetic abnormality in a single gene. We don't still fully understand the etiology. We think it's a mixture of genetic and environmental factors. and that there's the gut microbiome plays an important part. We think Western diet and lifestyle is important. Countries like China, which historically had low rates of IBD, as their lifestyle and diet has westernized over the last 30 years or so, have seen a big rise in the rates of inflammatory bowel disease. It obviously is a gastrointestinal disease, but it also causes extraintestinal manifestations, including joint pain, eye problems, skin problems, but also things like fatigue, and it can have a big impact on people's ability to live their life and get on with their work and so forth. So Clarity IBD is a study we set up last year. We set it up before vaccination was even sort of necessarily on the table. It was in trials, but we didn't know how successful those trials would be. And so our initial focus was on looking at what the impact was of the drugs, some of the drugs we use to treat IBD on COVID itself, so on having infection and the body's response to infection. We got going pretty quickly. This is our recruitment graph between October and sort of just around Christmas time last year, and we intended to recruit about just under 7,000 people by the 22nd of December, I think it was, and you can see we sort of pretty much met our recruitment target bang on, which was quite impressive, given that obviously it's really hard to predict these things up front when you're looking at such a big multi-site study. And we had 92 sites around the UK. Northern Ireland never got going, but we had sites otherwise across England, Wales, Scotland, as you can see on this map. It all happened pretty quickly. So our initial discussions were back in April, not long after I and others were involved in trying to map out what risk we perceived to be associated with inflammatory bowel disease medications and the risk of severe COVID. We then did a patient engagement exercise. We had about 450 people respond to an online questionnaire that we did as our patient engagement exercise, and that did help shape the nature of the way we ran the study, in particular with respect to returning results to participants. There was then a bit of time trying to secure funding and getting ethical permissions in place and so forth, but really relatively quick process through that, so that by September, we had urgent public health badging, which is what allowed us to recruit patients, because only urgent public health studies were being supported by the research networks for recruitment. And we recruited our first patients in late September. As I said, by December, we'd recruited 7,000 patients. And our first paper came out in March this year, and our second paper came out in April. I'll talk a little bit more about what I think enabled some of that rapid recruitment in a little bit, but I'm just going to talk through some of the results we've got to date. Some of these are published, others of them are sort of in the process of being published but aren't yet fully available. So one of the things, as I said, we wanted to look at initially was around what effect these medications, so infliximab and vedolizumab in particular, had on COVID and SARS-CoV-2 infection and the response to that infection. And so infliximab is both what are called biologic drugs. They work by targeting specific bits of the body's immune system. Infliximab is one that's been around for quite a while. It's first used in Crohn's disease over 20 years ago, and blocks something called TNF, which is part of some of the inflammatory pathways. And it's although it blocks that specifically, that's something involved in a whole lot of different sort of parts of the body, and is sort of regarded as having an effect on the body as a whole rather than the gut in specifically. Vedolizumab is a somewhat newer drug, and that works much more specifically in the gut to block white blood cells from being able to get into the gut when there's an sort of initial inflammatory trigger. And so it's generally felt to be a much more gut-specific drug. And in previous studies, vedolizumab hasn't been felt to have much effect on rates of infection, other than interestingly infections at the back of the nose, and also had been shown not to affect response to vaccines if they were given by injection, whereas infliximab had been shown to increase rates of infection and had also been shown to affect vaccine responses. So this is looking at antibodies to the nucleocapsid protein, which is part of the virus. And we can see there is a difference at baseline when people came into our study between the proportion of patients who had positive antibodies if they were on infliximab and vedolizumab. Now, that might mean, if you just had those data in isolation, that patients on vedolizumab were at higher risk of catching the virus. But actually, based on data from our study, but also from a number of different studies elsewhere, we know that's not the case. So the rates of PCR confirmed infection are about the same, and are probably about the same as the general population as well. So we don't think these drugs actually affect the rates of infection with the virus. So what we think we're measuring here is a difference in the body's response to the virus. So a difference in the proportion of people who mount a good antibody response against the virus. And of course, that may have implications for their risk of getting a further infection down the line. We could also see that if these patients were on, as well as being on infliximab, they were on an additional type of drug called an immunomodulator, so most commonly azathioprine, that had a further effect. So we can see there's further differentiation in the rates of having a positive antibody between those people on one of these immunomodulators with infliximab and those people who were not. And we can see that in the vedolizumab treated group, that's also true. So this 6% gets split out into 4.5 and 6.3%. And those two things are independently associated with a lower chance of having developed antibodies to the virus. And the reason it's important to look at that is because the more patients on infliximab are likely to be on azathioprine just because of the way we use the drugs. What about vaccination? Does this translate into a difference in response to vaccines? The short answer is yes, it does. So after a first dose of vaccination here on the left, we can see that actually the chances of getting what we defined as a positive antibody response to the vaccine was quite low in the infliximab treated group, so around sort of 20 to 30%, whereas it was about 60 to 70% in those on vedolizumab. So potentially quite an important difference there. Once people have had 2 doses of vaccination, actually things look a lot better. So regardless of which vaccine you had, so this is the Pfizer vaccine, this is the Oxford vaccine, and regardless of whether you're on infliximab or vedolizumab, you can see that the proportion of people with a positive response to the vaccine is well over 90%. However, there is still a difference. So about 6 or 7% of people, if they had the Oxford vaccine and were on infliximab, so these green bars here, failed to get an adequate antibody response to the vaccine after their second dose, versus perhaps less than 1% of those on vedolizumab. We also see that the immunomodulators that I mentioned before have less of an effect here than they did perhaps in the previous analysis looking at people who had the infection itself. T cells are another part of the body's response to the virus and to vaccination. This is a rather complex slide, but the short version is there isn't really a difference in T cell responses between the 2 drugs, and also that there was a relatively poor association between the antibody responses and the T cell responses. So these 2 aspects of the body's immune response to the virus seem to be somewhat decoupled. So some people will have a good antibody response and not so good cellular response or T cell response, and in other people it will be the other way around. But there was a weak association. I guess the other thing that we might be concerned about, particularly if the antibody responses are a bit lower following a second dose, is what happens if we take people out to 4, 6, 8 weeks and on 6 months. And so these data predate the introduction of third doses or booster doses, and we can see that the orange line, which is the vedolizumab treated people, whether they get the Pfizer vaccine or the Oxford vaccine, they get a bit of a rise here with their first dose. It then sort of plateaus out a bit. It then goes up a lot more after their second dose, and then slowly drops away. And this purple line, these are data from a different study run by UCL, looking at other people who don't have inflammatory bowel disease and aren't on these medications. I think broadly speaking, I would say that the purple line and the orange line are about the same. And given that there may be different, though it's the same assay, there may be differences in the way the laboratories are set up. There probably isn't much of a difference here. The green line, though, which is people on infliximab, you can see not only is the the peak both after first dose and after second dose lower, which is what I showed you on previous graphs in a slightly different way, but they also probably drop away more quickly, and they drop rather more quickly down to this blue line here, which is where we were regarding people as having had an adequate response to the vaccine. And we can see with the Oxford vaccine on the right that the peak response is a bit lower, and possibly the decay then is a bit slower, but they do get this lower peak antibody response. And that doesn't necessarily mean that they're less well protected, but obviously it is a potential concern. This is another way of looking at those data where we've got a line for every individual patient in the study. And you can see that generally people go down after their second dose. You can see that they probably do go down a bit quicker. So the half-life is shorter for people who are on infliximab than on vedolizumab. Again, that means that we would have concerns perhaps that these people on infliximab are going to be at greater risk of sort of breakthrough infection. So do we actually see a difference in breakthrough infection? Well, we do. So before vaccination, as I said, our study and others have shown there's really no difference in the rate of infection between people on infliximab and vedolizumab. And so these sort of cumulative proportions of people with an infection prior to their first dose of vaccination are more or less the same. If we look at people after they've had the second dose of vaccination, of course, some people will be on both graphs because they'll have been on this one before they got vaccinated and this one afterwards. You can see we do start to see separation between the lines. People treated with infliximab have a higher cumulative rate of infection than people treated with vedolizumab, suggesting that given there wasn't a difference beforehand, it probably relates to vaccine responses. We've, of course, now lucky enough that we have booster and third doses, and there's obviously a bit of confusion about those two, but for most of our patients, actually, their third dose, which they were eligible for, came about 6 months after their second dose anyway, just because of the timing of the rollout of the vaccination program. But what we can see here is this is an updated version of the graph I showed before, is that once people have had their third dose, both in the vedolizumab and importantly in the infliximab treated groups, we see a nice, even higher rise in antibody concentration. Now, although they had different vaccines at baseline, so this is the Pfizer group at baseline and the Oxford group at baseline, pretty much everyone had Pfizer for their third dose. So this is probably seeing that we're seeing a similar rise actually, regardless of which vaccines you'd had before, up to well over 1,000, which is really good. So I think Clarity has been really successful. Our data has been used by the CDC in America, and I think has influenced policy in Canada and the UK as well around third doses and booster doses, and has had an impact certainly beyond inflammatory bowel disease. I think in terms of how we've been able to deliver this study, we used lots of e-consent. It was the first big study I've been involved in running where we used e-consent, and that was generally successful. A lot of that was in-person e-consent on sort of tablets in the infusion suite, but we've also done remote e-consent as well during this study. We have had lots of data. At last count, we've had over 9 million data points from our participants, and most of that's come directly from the participants themselves using electronically administered surveys, although some has come from the research sites. We, as I said, influenced by patient engagement prior to opening the study, we've been returning the serology results directly to participants, so they've been able to see whether they've got a positive antibody test, either the one that only goes up if you've had the infection, or the one that goes up either with vaccination or infection. And we've been responsive to changes in the pandemic as well. So vaccination was something we hoped would happen when we were setting this study up, but we really didn't know, and we didn't know what the timescale would look like. But we were able to quickly pivot from a study looking at infection to one that really now is, I think, more importantly, focusing on vaccination. We've been looking at long COVID. We've got some data on that at this stage, which is unpublished and not sort of ready to share, but we will have something to say about that. And we're also looking at persistent viral carriage. So one of the concerns is that if you don't get a good antibody response to the virus, maybe that means you'll have persistent virus up your nose or elsewhere, even once you've otherwise recovered. And maybe that's an opportunity in those people for further mutation to take place in the virus. And there's been some suggestion, not in our patient population, but in other patient populations, that that has happened and may have contributed to the emergence of some of the new variants of this virus. So that's something we're looking at as well. So far, actually, the story there seems to be pretty reassuring. We've only had 2 people who've had a positive PCR following their initial one, and they've both been reasonably close to their initial one, where we might expect that to be the case. So hopefully that's not going to be an issue for our patient population. So I think in conclusion, some of the biologic medications that we use in IBD can affect vaccine effectiveness. We can use 3rd doses to overcome this. And also, really, this has been, I think, a real success story of delivering research at scale through the pandemic, in collaboration with lots of people around the country, also in collaboration with participants who we've engaged with a lot directly, and making good use of electronic tools. We've tried, where possible, of course, not to exclude people if they can't use the digital tools, and so we've had other means of engaging people. But I think it's, as I said, been a real success in how we can use technology to drive research. So that's all I wanted to say sort of formally just now, but I'm really happy to answer questions. We've got quite a lot of other data that I haven't presented here, which I may be able to draw on if I need to. But if anyone has any questions, now's an opportunity.
Thank you, Nick. That was a fascinating presentation, and I'll just open it to the floor for questions. So while people perhaps having a think about some questions, it's amazing. The recruitment rate that you did in such short space of time was phenomenal. That's absolutely fantastic.
Thank you.
And I could just see Bob just put your hand. Bob just put his hand up. Have you got a question, Bob?
Yes, I gathered from what you said that you passed the information on the whatever the right word is, the amount of antibody,
yeah,
to the participants. To what extent would this have affected their behavior and their results, do you think?
So it's a good question. I think in terms of, so we did look at things like shielding behavior and so forth, and one would imagine that might have been influenced by what we passed on to participants. I don't think it would have affected the antibody results themselves, because other than the fact it may have, I guess, might have encouraged some people to take up a 3rd or booster dose earlier than they might otherwise have done. But I don't think it will affect any of the key results from the study. I guess in terms of breakthrough infection, if there had been a situation where people who were told they had a positive antibody were likely to engage in riskier behaviours that put them at greater risk of acquiring the infection, we'd expect to see some muting of the effect we actually saw. So we'd expect, if the antibodies didn't matter, we might have expected actually the vedolizumab treated group who had higher antibody concentrations to have more infections, because they were perhaps behaving in a riskier manner. We have also always been very cautious in the way we present these results to say, this is what they are, but we still don't know really what this means in terms of your individual risk. And because we've got obviously engagement at the local sites from research nurses who can field questions, but we've also got a team in Exeter who've been, including me, who've been directly interacting with some of the participants when they've had questions. I think we've often been able to sort of, I guess, anticipate some of that and make sure that the information is fed back sensitively. We've also shared newsletters with participants with the sort of key results, and we've had we've had one video out so far. We've got another one in development as well, which is aimed at participants rather than other colleagues. And to the general public. It's not just for participants. If people are interested in the first video we had, I can share a link to that. It's on YouTube.
Can I just come back on that? I mean, one point I noticed was that in some cases, the levels of antibodies dropped almost to zero, or were almost zero. And if I were in that position, I would be worrying. Yes. And trying to get another vaccine or whatever might be appropriate. Well, how did you handle that?
So there were very few people dropped actually all the way to zero, but people did drop below the threshold that we were calling as positive within the study. And other than when we had specific requests, we weren't showing people the numbers, we were showing them whether they were above or below that threshold. So yes, that certainly did cause anxiety in some participants. we obviously, we do believe that the level of antibody concentration, we've got some data to support this, does correlate with sort of protection from the virus. And so we did say that to people that, if they were low, it's possible that would indicate they were at higher risk of further infection. But also we sort of emphasized that it wasn't a sort of an exact thing where people with high antibody concentrations would never get it and people who were low were very high risk. So I think it probably did. It probably did generate some anxiety. We've had one person who explicitly opted out of wanting to know if they'd responded, but that was 1 out of 7,000. So that was pretty unusual. And I think in general, people were keen to have access to that information. When we did the survey prior to when we were planning the study, I think out of 400 or so people who responded, we had one who said that they wouldn't want to know their results, even if we didn't know what they meant.
Right.
So it was, I think it has, and I think it's definitely helped with keep people engaged in the study. People email me effectively, usually if their results haven't come back within a couple of weeks of them having their blood sample taken, saying, where's my result? People sort of want to know.
And Austin, I see you've got your hand up there.
Yes, thank you. Fascinating study, and most difficult to do it in the circumstances of an unprecedented pandemic. But what occurs to me, which I appreciate is probably outside your own discipline, is whether presumably there were parallel studies for drugs in other disciplines. I do remember at the beginning, fairly near the beginning of the pandemic, there was much concern about the use of ACE inhibitors and the other similar drug, the ARBs, as to what effect they were having in relation to the pandemic. So I'm interested to know whether you're aware of how many other disciplines were running studies that were kind of parallel with yours.
Thank you. That's a good question. So in terms of drugs used in hospitals, like the ones that I've talked about, there are, so Octave is a study that was, I think, led from the University of Birmingham that recruited about 5,000 people. That was looking at vaccine response to the vaccines for SARS-CoV-2. And they were looking at a range of immunosuppressed groups, including liver transplant patients and people with HIV, I think, and various other things. And they have reported in some of their initial results. They got more press than we did, perhaps because it was across many diseases rather than one. In terms of looking at drugs that are perhaps more commonly prescribed in primary care, probably the best place to look at those data are some of the big national data sets that have emerged during the pandemic. So Open Safely is probably the best known of those. I'm not sure if they've looked specifically at ACE inhibitors and ARBs. It would be an interesting thing to look at because of the fact that the virus does bind to ACE. And I am involved in some other open safety projects, but I'd have to look into whether that's something they've ever looked at. As far as I'm aware, no one's reported that there is a strong association or even an association with ACE inhibitors or ARBs, but I'd need to double check that because it's slightly outside my area.
Thank you.
Okay, so Alexis.
Sorry, Kim.
No.
Hand up a long time. My question was really the same as Austin's as to, but I've just changed it slightly. Your design, is it unique, or have other people taken the research design and applied it? And have you written about the lessons learned and what you require, like, fantastic people who gather 7,000 and keep them going and all that stuff? What? Because it's very impressive, your design. I can't understand any of the graphs, but never mind about that. It's the way you worked it. So is that being translated?
So I think that there are other studies that have looked at and that have innovated during the pandemic, and some perhaps even more impressive than ours. So Tim Spector's Join Zoe COVID-19 project, which I think originally came out of Twins UK and was going to be something when you used to study twins and COVID, but opened it up to sort of millions of people in the general population, I guess is a really good example of something that's been run at a national level, and that predates our study. So they certainly wouldn't have taken inspiration from us, perhaps more the other way around. I think I said I don't know much about the way Octave ran. But it was a similarly large study, although I think perhaps a better funded one than ours. I think in terms of us disseminating what we've done, we've been talking about that within the gastroenterology community, but we are planning to write that up. We've been asked by NIHR to write up our, what we've learned from it from a delivery perspective for their sort of I think it's a magazine they have rather than the journal, and that's so that will be available freely, I think, on their website once that's done. But we're also looking at maybe having a formal peer-reviewed publication in the scientific literature on that as well. So yes, I think it's I think everyone is being forced to sort of modify the way they do things. It's perhaps just accelerated what would have happened anyway, but I'm involved in another study that's looking at interventions for diarrhea in ulcerative colitis, and that was originally going to be delivered in person, and it can still be delivered in person, but it's shifted, partly through my involvement, over the last year to a study that can be fully delivered remotely, including remote e-consent and remote delivery of dietetic advice, which is part of the study design. So yeah, so I think we're one of a number of studies that have been innovating, and we are aiming to share that learning with whoever's interested in reading it.
Brilliant. Thank you so much, Nick. That was a fascinating talk, and I'm really thank you so much for coming away from your clinical time to come and talk to us. We really appreciate it.
No worries. Slightly ironically, I'm actually self-isolating away from the ward at the moment because one of my family members she's well, but has a positive PCR. So I would normally have been actually breaking away from being in person on the ward today, but I'm having to call from elsewhere. But it's really interesting to speak to you, and I'm happy if there are questions that come up after this. I'm happy to, if people need my email address, whatever, I'm happy to try and answer those.
Brilliant, thank you. We really appreciate that. And we'll pass anything on to you. So I'd just like to draw this session to a close because we're going to go into a lunch break now, and we